So now I will start with the presentation for today's presentation for rare diseases and the genetic technologies which are used to detect these rare diseases. So before moving to the presentation, I would just like to give you a brief of DSS Imagetech uh, and its contribution to life sciences uh, division and its contribution in India. So DSS is the leading advanced diagnostic company in India. It was established in 1998 and it is backed by an 80 years legacy. DSS Imagetech delivers cutting-edge life sciences and healthcare solutions with the philosophy that it follows inspired by technology and Make in India mission. DSS also ensures excellence through end-to-end technical application and validation support across all routine and specialized testing platforms. As you can see on the screen, DSS operates with multiple divisions under it, such as microimaging, MI division for microscopy, clinical diagnostic division under which there is histopathology and molecular, in vitro fertilization division, IVF, food testing division, and medical division. So now this company operates across all these sectors and then combined, it provides a great solution to Indian market and Indian population in the life sciences sector. Now we will move to today's presentation. So to give you an overview of today's session, we are going to start by defining the global and Indian landscape of the rare diseases. We will then look into the current national policy, which is NPRD 2021, which is the major pillar for the patient support in India. Then, crucially, we'll also dive into the diagnostic odyssey. It's the long journey that patients face, a rare disease patient, and then how molecular technologies such as MLPA, NGS, they are now closing this gap, and it is bringing us to the new curative era for rare diseases. Okay. Now, in discovering rare diseases, let's start with the important point. What is rare disease? So, rare diseases are chronic, debilitating conditions that affect very small percentage of population, but yet collectively they impact millions of people worldwide. While this definition vary globally, whereas WHO classifies it as a disease which is rare when its prevalence is less than or equal to 1,000 individuals. Some countries have stricter or tighter thresholds, such as less than or equal to 1 per 2,000 individuals. Now, despite their individual rarity, these conditions present a massive global healthcare challenge. And because they are often lifelong and they also require specialized medicines and social support. Okay, moving to the global rare disease landscape. Rare diseases are complex, they are multi-system disorder, and they are characterized by extreme phenotypic heterogeneity, making them difficult to diagnose and manage. Now, while we already saw, WHO defines it as a condition which affects less than or equal to 10 people per 1,000 people. Nationwide, these definitions vary significantly so it reflects that the local healthcare priorities are different in different countries. So for instance, the threshold is very tighter in Europe, which is less than or equal to 5 individuals per 1,000, and even more specific in USA. So where now it is defined by the total population count of fewer than around 2 lakh people. And in developing countries like India now, there is a lack of standardized definition. And also there is lack of epidemiological data, which remains a critical barrier to quantify the true burden of these diseases and their associated mortality in India. So, what do you think for countries like Australia and Taiwan, as mentioned in this table, they have a threshold of just 1, while US has it as 6.4. So, is it just about the science or a budget? So basically, it's both, because a definition is not just a number, it is commitment to fundings. Because as we move to the next slide now, we need to keep in mind that definition of rare disease in India remains invisible for millions of people who are suffering from rare disease. Okay. Now moving to the moving to the rare diseases facts. Rare diseases are individually very uncommon, but when they are collectively together counted, they affect millions of people worldwide. With nearly around the ones which are noted are around 7 to 8,000 rare diseases identified today. So around 70% of these rare diseases have onset in childhood, and around 72% of the rare diseases have genetic origin. Hence, it makes the molecular testing very essential for diagnosis of rare diseases. Now, patients often experience an average diagnostic delay of about 5 years due to multiple specialist consultations and also incorrect diagnosis. But as these diseases are rare, these diseases affect around 300 million people worldwide. So currently, about 5% of the rare diseases have approved targeted therapies from US FDA, which highlights a significant unmet medical need and importance of genomic diagnostics in improving patients' outcome. So if we count 95% of these 8,000 diseases, they lack the targeted cure. Does this like, is diagnosis for rare diseases important still? So answer is yes, because for a family, diagnosis is end of this 5 years odyssey, the long journey that they suffer from, and also the beginning of informed supportive care and clinical trial eligibility. We'll just go to the definition of rare disease in Indian scenario, how we see it in India. So rare diseases are conditions we saw that they occur infrequently in population. But in country like India, collectively they are significant. So as per estimate, it is around 70 to 96 million people who are affected by rare diseases. If we consider the global prevalence estimates of roughly 6 to 8%, and so in India, the major challenge, as we see here, is the underdiagnosis due to limited awareness. So also there is less limited diagnostic access, and also variable definitions are based on prevalence of the population. So India does not have a single universal numeric definition, but yes, it does recognizes rare diseases as a condition that occur infrequently and require specialized diagnostic and management approaches. Now, this emphasizes the need for molecular diagnostics and a structured rare disease program to support early, accurate diagnosis. We just quickly see rare disease in Indian context. So, in India, rare is a misnomer because due to massive population, we are just not dealing with the isolated cases. We are dealing with the millions of citizens which are scattered across the entire geography of India. And for many of our patients, diagnostic odyssey is prolonged by uneven access to molecular labs. So for a child in rural village, he might have to wait for like long period as compared to the child in metropolitan area to get the rare disease diagnosed and to get the treatment. As you can see on the screen, multiple rare diseases are just mentioned in this map. Okay, now we move to the National Policy for Rare Disease, which was established in the year of 2022. So we think of what happens if the disease is very rare, it is expensive to treat, and also difficult to diagnose. So this is the exact problem that is being addressed with this 2022 National Policy for Rare Diseases. Later, it was strengthened in 2022 by the financial amendment, where the funds which were given for the treatment of the patients were increased from 20 lakhs to 50 lakhs. So first is the national framework that is designed under this policy. So instead of treating all the rare diseases in the same manner, the policy categorizes them into three groups based on the treatment complexity and the cost which is involved in them. So why it is very important in territory like India? Because resources allocation in country like India is most strategic, equitable, and evidence based. Now let's we move to the center of excellence. So now imagine if a patient finally gets referred to a specialized center. So not just for the treatment, but for the accurate genetic confirmation. Here comes the center of excellence in picture, because government, these are like government designated hospitals which are equipped with advanced genetic, molecular diagnostic, and multidisciplinary care pathways. So they have all the solutions to get the testing for rare disease done for the patient. Also, as I already mentioned, the financial support was updated in 2022. It is so finance and funds are the major barriers for rare disease affordability, as the treatment is very costly. So originally support was just limited to 20 lakh. In 22 it was expanded to 50 lakhs, and but it is only applicable for the rare diseases for which the center of excellence is going to do the test, the molecular testing. So once the molecular diagnosis is confirmed, the patient becomes eligible for the fund for the treatment. So this is a diagnostic mandate, no genetic confirmation, no funding. So molecular diagnosis are not just clinically important, they are also policy enabling now. Okay, next we'll move to the very important topic that we just spoke about in rare diseases, the diagnostic odyssey, or the long journey that the patient goes through during uh, their entire journey of the rare disease suffering. So most of the families, they don't just walk a straight line to a cure directly. They fall into a river which we call as a river of uncertainty, which you see on the screen. So this journey begins with the vague symptoms that lead to the cycle of long waits. There is testing cost involved. Multiple times there are misdiagnoses which are happening. So there is a specialist referral gap in this diagnostic odyssey, because even after reaching to the neurologist or geneticist, the path remains precarious until the molecular tool is applied here. So this is where this 5 to 7 years delay happens. It is not because of the lack of effort. It is because the lack of right definitive test that needs to be done. So, what is the exit strategy for the diagnostic odyssey? It is genetic testing. So, by moving this step earlier in this entire process, we can avoid this uncertainty river which swallows the like multiple years of that child's development, and we can move directly to accurate treatment. In 2022, the mission is to make this treatment part as the treatment journey as less as possible. So we have just seen about the diagnostic odyssey that the child goes through around 5 to 7 years. Now here we see that most of the families are again trapped in uncertainty river. They are just bouncing between the initial signs, misdiagnosis. So during this time, they often go to 6 to 8 different specialists, maybe around 17 hospitals at that time. So it is not just the delay, but it's an emotional, financial, and medical exhaustion to that patient. Okay. So also, yeah, so we can quickly take this example here on the screen, which is for the DMD. Symptoms begins at 2 to 3 years of age, but the genetic confirmation happens at 4 to 6 years. So there is a gap of 2 to 3 years here. Okay. Now we'll address the zebra in this entire journey. So let's say if we close our eyes and we hear, we are standing in the field and we close our eyes and we hear the hoofbeats. The first animal that comes to our mind would be mostly an horse. right? So why this happens? Because this is general concept. In medical schools also, the students are taught that when you hear the hoof boot, you think of horses, not the zebras. So it is the rule for efficiency. Look for the common cold, common diseases rather than a rare mutation. But today, I would like to talk to you about those 96 million people in India for whom those hoofbeats are zebras. So for a rare disease patient, thinking horses first, it is a big nightmare. Imagine a family who is waiting for 7 years and they are seeing multiple specialists and still they are not able to reach to any conclusion. So while here doctors are looking for the horses, the zebra is right in front of them and the disease is also progressing every single day. So this is why rare disease community has reclaimed this zebra label. So it is no longer a sign of being missed. It is just the symbol of strength and uniqueness. So just like two zebras, they do not have same strips. No two rare diseases in this journey are alike. So they are reclaiming the right to be seen with this symbol of zebra. Now, the reason that we are presenting on rare diseases is this is the rare disease month and we have just now discussed about the zebra journey. Every year the last day of this February month is something something which is not spoken always is done, which is rare disease day. So it is not just the day on the calendar, it is the day where the world stops and thinks of rare disease particularly. So this year, this movement has also shifted and from awareness to equity. But why we are talking about equity in rare diseases? Because awareness without diagnostic tool, it's just a frustration. So true equity means a child in tier 2 city of India has same access to techniques, molecular techniques like MLPA, NGS as the child in Europe. So that is the gap we are here to close. Today, as we observe Rare Disease Day in this month, we will join the global chain of light. So across the world on this day, hospitals, different landmarks, they are illuminated with different lights such as pink, green, and it signifies that the no patient should remain in dark in this entire journey. Now, this movement has really grown so much that around more than 6,000 events across more than 100 countries. It is celebrated and it reflects the massive international scale that the rare disease has got. Now we talked about rare disease, but we need to also understand how to analyze these diseases and what exactly the rare disease testing is. So when a child or a patient shows unexplained symptoms that don't even fit into the common conditions that are usually seen, what do we think the doctor will do next? So here comes the rare disease testing part. Rare disease testing involves high resolution molecular cytogenetic analysis. So in simple term, we look at the patient's DNA very closely to find the changes like a single letter change in the genes, missing or extra copies in the DNA, or even chemical modifications such as epigenetic changes, methylation. So, what is our aim here? Our aim is to end the diagnostic odyssey at the end because there is a long, frustrating journey that the patient goes through. So here we'll just quickly look into the genomic context also. Human DNA has around 3.2 billions of letters, the nucleotides, and around 1 to 2% codes for protein, that is the exome. However, changes made anywhere in the entire genome can cause the disease. Now, in the genome, what causes the rare disease? So you can see different variations mentioned on the screen. These are something that we look for with this genetic rare disease testing. Single gene mutation, single nucleotide variants, copy number changes, chromosomal abnormalities, epigenetic alterations such as imprinting defects and methylation. So these variants can either be inherited from parents or they occur de novo. That means they appear for the first time in that patient. So sometimes understanding this origin of the disease, it helps the family further for the counseling and also to do the risk assessment. Okay. Now we'll move to the precision diagnostic part, which is the backbone of NPRD 2021. So let's start with the most critical update of 2022 or 26. Funding for rare diseases has, as I said, it is actually already doubled in the year of 22. Now the grants went to 50 lakhs. So, but in this entire thing, you can see NPRD has prioritized certain diseases, and the policy has mentioned certain rare diseases. We already discussed about the 70 million people which are affected in India. It is the estimated number. So here I just want to tell you there is a big misconception for fundings that are done. We think that the fundings are only for below poverty line families, but that is not correct. There is no restriction for BPL, and anybody who has the rare disease diagnosed at the cancer center of excellence is eligible for this. And how can we unlock these funds? So the answer is again genetic confirmation. Government will only release these funds for a molecular diagnosis which is validated by the cancer of excellence. Moving to the next slide, for key types of genetic variants. So now if we need to navigate from the uncertainty river, as we discussed earlier, we must understand the language of the genome. So it means the mutation range, it ranges from single typo, such as SNVs, to entire missing chapters, such as copy number variations. So SNVs are single nucleotide variants. They are critical in sickle cell anemia. We also see indels, which is insertion and deletions. Indels are very small insertion deletions, less than 50 base pairs, which are commonly seen in cystic fibrosis. Next is large scale changes such as copy number variations and structural variants. These are critical for many patients. So, for example, in conditions like SMA and DMD, exons are deleted, large segments are missing and duplicated. Now, because this involve like large chunks of DNA, they often require different testing tools than a simple point mutation testing. So, we also see here UPD, which is uniparental disomy, where a child inherits both chromosomes from just one parent. It leads to a disorder like Prader-Willi. And then you can also see LOH, which is loss of heterozygosity, which is the primary driver in pediatric cancers like retinoblastoma. So why this matter? Because if you suspect a DMD, then a test designated by the SNV will come always negative. So your patient will stay in that uncertainty river for quite long time, and you must match this test to the variant type that is associated with the disorder. Okay. So regarding molecular diagnostics in rare diseases, so we have just visualized the uncertainty river. So it involves the process of advanced technologies. So here you can see application of advanced molecular and cytogenetic tools are used to identify the pathogenic variants. We just spoke about the different variants that are there. Then and also this particular circle shows the different variants which are involved: SNV, small indents, CNVs, methylation, and structural variants. So this one gives a definitive genetic confirmation. So why this is important again? Because this will end the diagnostic odyssey, the long journey that the patient goes through. It will enable the targeted therapy selection. It will also support for the carrier and prenatal testing. And also these tests will help us to identify the prognosis and recurrence risk of these diseases. Talking about the cytogenetic approaches in rare disease investigation. So first, what is cytogenetics? Cytogenetics is the study of chromosomal structure and numerical abnormalities to identify the genetic disorders. Which are the core techniques, major techniques which are covered under cytogenetics? It's karyotyping, to check the structural changes and chromosomal deletions. Then fluorescence in situ hybridization for targeted loci detection. And then chromosomal microarray. It is again for copy numbers. As compared to karyotyping, it has a higher resolution. So as we said already, the capabilities range from aneuploidy, large deletion duplications, also microdeletion syndromes, and also to detect balance rearrangements. Talking about the resolution, especially for karyotyping, it has a resolution of around 5 to 10 MB, whereas CMA has much higher resolution as compared to karyotyping. Okay, now we'll talk about the genomic sequencing. So if cytogenetics was about looking for the bookshelves in a particular library of our DNA, then genomic sequencing is about reading the actual text on each page. So this technology has really revolutionized our field by allowing us to identify smallest of the typos or nucleotide changes that are there in the DNA, which were once invisible to us. So whether we are looking at a specific set of genes or the entire chromosome, we can detect the changes. So how this generally works? So it works with these technologies mentioned on the screen. First is targeted gene panel. So it is a focused area which is for the known disease causing genes, and where you will see the targeted panels which are involved for your area of interest or area, gene of interest. Then second one is whole exome sequencing. So here it is just reading only the protein coding regions. So about 2% of the genome is exome, whereas about 85% overall disease, that rare disease or diseases that happen, which happens in the exome region only. Talking about the whole exome sequence, whole genome sequencing, it is most comprehensive view of the genome because it reads everything from exome to non-coding regions in the DNA. So it's the complete read of your genome. Now, here the strength of this NGS, it lies in its incredible resolution, because we are not here looking for the missing chapters, but we are exactly looking at very small level detections such as SNVs, single nucleotide variants, small insertion deletion less than 50 base pair. Also in cases, some CNVs can also be detected with genomic sequencing. Talking about the advantages of this, the primary advantages is broad coverage, which is combined with the high resolution. Now, as such, no technology is perfect. So, when we are understanding the limitations of this technique, NGS still can miss the exon level CNVs, the balance rearrangements, and methylation defects, because it does not tell us like how the genes are turned on or turned off. We'll move to the genomic iceberg in rare diseases. So we often think that whole exome sequencing and whole genome sequencing is the end of the journey, but it is not the complete truth, because even after most of the advanced sequencing, 30 to 50% of the cases remain unresolved, and this is what we call as 50% gap. So now you can see, look at this iceberg. What we see above the water are SNVs, single nucleotide variants, the single letter type O. That the standard NGS is great at catching, but now the bulk of the mystery lies in the massive hidden depths which are below this water surface. So now if we look at the hidden part here, you can first see CNVs and structural changes. These are large scale, again we have already discussed about them, large scale deletion duplications, which, like, if you go by sequencing, it will often get missed. Second is methylation. These are epigenetic regulations where DNA itself is normal, but there are switches which are broken. So it is the DNA is normal, but there are external factors which are controlling its activity. And then there are complex regions such as repeat expansions, chromosomal rearrangements, and there are also non-coding sequences, which whole exome sequencing also may not read. Now, to solve these cases, we have to move like from this shallow region to this deep region, because here integrating transcriptomics with a specialized structural pathway becomes important to identify all the hidden unknown mutations that could be there. Some mutations may be to find out the rare diseases which are not yet discovered. Now, one more key part that this diagnostic odyssey happens, and one of the challenges in the rare disease genetic testing is the clinical bottleneck. Now, we will address this particular term called as variants of unknown significance, VUS. Now, this is a very gray zone because it creates like a bottleneck for the clinical practice generally. So, it is a challenge because finding out variant is often just the beginning of the journey. I mean, it is not the end, because we'll just explain that through these points. First point is determining gene disruption. So here, just because we identify a change in the DNA doesn't mean that we have found out the cause. We have to prove that the specific variant is actually one which breaks the gene's function before it can be used to guide the clinical decision or for the treatment purpose. Now, next, we are seeing distinguishing from the non-disease variant. So what is this? So every individual has around thousands of genetic variations that are perfectly normal. So our task here becomes to identify the main disease causing variant in this all background noise. The third one is non-coding variant here. So these are not in the gene themselves, but now they act as the regulatory switches which turn on or turn off the genes. So if we don't count for these, we might miss the diagnosis entirely, even if the gene itself looks fine. The next one is undiscovered disease genes. So we are working at the very edge of the medical science here, and often we identify mutations in the genes that are not even documented in the textbooks yet. So it requires deep expertise and to link these novel genes to the patient's symptoms. The last part is analytical limitations. So, as we already spoke, no single technology is perfect. Standard sequencing might miss complex structural changes, deep intronic mutations. So, we must constantly refine our tools to ensure we are not missing the truth due to the technical blind spots which are there in rare disease testing. We'll move to the next part for of integrating these diagnostic technologies together. Okay. So now we have just seen this iceberg with the shallow and deep regions of having different variants. And we have also understood no single technology is the magic bullet here. The future of rare disease diagnosis is not about choosing one test. It is about integrating them to leave no stone unturned for the actual diagnosis of the rare disease. So as you can see, first one here is sequencing. So sequencing, it remains a very great method to identify the SNVs and small indels. MLPA, it is great at exon level deletion duplication testing that the sequencing might miss. Cytogenetics, it is a macro lens here because it is for the structural rearrangement detection. MSMLPA, of course, it's part of MLPA only. It's another key test to detect the methylation defects and epigenetic regulations. So the question is why we are on this point of integrating. Why to integrate? Because, for example, NGS, when it is combined with MLPA, it will significantly improve the detection as compared to the NGS test alone. So it will definitely transform. Let's say there is a likely negative result. It could also turn into the confirmed positive if we combine multiple tests together. So in a clinical settings, we need to use a tiered diagnostic workflow what it will do, it will first help in the balancing the cost, speed of testing and diagnosis, and also it will be very comprehensive as compared to single test. So as we start with the complementary methods, we can easily fill these gaps of diagnostic odyssey early. There's one important term here, which is multiomics and integrative tools, which should be involved here. They are future for the diagnostic precision in rare diseases, because it integrates different sectors such as genomics, transcriptomics, proteomics, metabolomics. So this helps us to overcome diagnostic, prognostic, therapeutic, and many other hurdles that are there. So by combining these different layers together, clinician can better interpret the variants of unknown significance we just discussed. Moving to the next slide, of importance of copy number variation entire scenario. So we often focus on the missing mistakes in spellings, okay, in the DNA, but the copy number variations, they are like missing or extra pages of the genetic manual. So what are they? They are large scale deletions, duplications, which are seen in the DNA, and they are very fundamental to the molecular basis of many rare disorders. So you can see CNV contributes to the molecular basis of many genetic disorders, and which are those? So they are neuromuscular disorders, developmental delays, imprinting disorders, and so on. There are many diseases which are covered under this. So here, large clinical exome studies have shown that incorporating CNV studies increases the overall diagnostic yield. And to achieve the maximum diagnostic yield, it is CNV analysis is no longer optional. It is very essential for identifying the root cause of this congenital and neuromuscular conditions. So we'll talk about what is MLPA. So MLPA is the patented technology from MRC Holland. It stands for the multiplex ligation dependent probe amplification. It's a molecular technique. It is used to detect copy number changes. It is considered as gold standard here, and copy number changes such as deletion, duplications in specific region of DNA. So why MLPA is valuable? Because MLPA it shows high sensitivity for the targeted CNVs. It robustly detects CNVs, which are often missed by sequencing. It also has a multiplex capacity. So for say, for one sample, simultaneously you can detect up to 60 targets. It is rapid and cost effective because it starts from single reaction, PCR happens, fragment separation is done, and then you analyze the result on Coffalizer software. Mm, okay. Okay, Coffalizer software within 24 hours. So, talking about the clinical utility in rare diseases, MLPA becomes the primary diagnostic tool for DMD and SMA. Here, DMD stands for Duchenne muscular dystrophy, and SMA is spinal muscular atrophy. We'll talk about these diseases in detail later through the presentation. Now, talking about MSMLPA, again, this is used for detection of copy number and methylation abnormalities. So, what is MSMLPA? So, MSMLPA stands for methylation specific multiplex ligation dependent probe amplification. It's the variation of MLPA that detects both copy number changes, deletion and duplication, and methylation abnormalities, which are epigenetic changes. Both happens in a single assay. So it combines copy number and methylation analysis in one assay, as I already said, and it is effective with small amount of DNA, high diagnostic value it has in suspected imprinting disorders, and also there is lower cost and fast turnaround time, less than 24 hours from sample to results. So which are the primary targets here, or the primary rare disorders under this? So MLPA or MRC Holland provides a kit for Prader-Willi and Angelman syndrome. So it identifies the deletion in 15q11, q13 region, and also it shows the methylation patterns present in this. So in short, MLPA bridges the genetic and epigenetic gap in molecular pathology. Okay, talking about the tiered diagnostic workflow in rare diseases. So here you can see tiered testing is essential, and to minimize the again diagnostic odyssey to get the confirmation of the rare disease as early as possible. So generally first basic thing is done is phenotypic assessment of the patient along with some biochemical testings, and then it is followed by the structural and targeted molecular tests such as cytogenetics, karyotyping, CMA, fish. Also, it involves MLPA and MSMLPA for copy number variation and methylation assessment. Then next step where one can move is tier 2 testing. Here targeted NGS panels are included. It is for the phenotype driven gene panels. It also detects SNVs, small indels, and selected CNVs as we already discussed. The next part of this tier testing is comprehensive genomic testing. This is the last one where whole exome sequencing, the protein coding region it is sequenced, and whole genome sequencing where coding, non-coding, everything from the genome is sequenced. So why this tiered approach is important? It improves our diagnostic yield by large percentage. It also improves the cost efficiency during this entire diagnostic journey of rare disease patient. It reduces the turnaround time for the testing, and it also increases the accessibility in resource sensitive settings, maybe some rural parts where there are less resources available in that setting. Okay. Now we will talk about MLPA. So we have already discussed about MLPA, but we will just see what MLPA is about. So MLPA, it's a gold standard for copy number variation analysis. It detects the copy numbers using PCR-based method. So first PCR is done, and then these amplified fragments are separated using capillary electrophoresis device. It's just the two devices which you need, which are I think available in most of the labs across India. So this tests up to 60 genomic regions in single reaction. So high multiplexing is there. It uses a single universal primer pair with a very high sensitivity. So there is no amplification bias, which is generally seen with the multiplexing. And it also distinguishes the target region from a very similar pseudogene in case that is present with a single nucleotide specificity. So this happens because of the ligase enzyme that is involved in this kit. As we again already spoke, the workflow is from sample to result within 24 hours. The gold standard diagnostics. So the major two rares in there are many products or MLPA kits which are for the rare diseases, but the major two are the DMD and SMA. Because for this DMD, it becomes the first line of diagnostic test. And what we provide with this is SALSA MLPA probe mixes such as P034, P035, that are like long-standing first-line tests, and it detects about 70% of the copy number variations. SMA, again, it's a gold standard. We have products such as P021 and 60 to quantify the SMN1 gene, and also it discriminates SMN2 from SMN1. So in short, this technology simplifies, streamlines the identification of genetic deletions and duplications, which are essential for early intervention. So talking about the workflow, you can see on the right side, it starts from denaturation, hybridization, up to data interpretation. So there is overnight hybridization involved here. Everything happens within 24 hours. Okay. Now talking about the DMD part, this is Duchenne muscular dystrophy. Again, MLP is gold standard, as I already mentioned. So DMD is one of the devastating rare disease we face. And so what happens in DMD? So there is a germline defect in this gene, which is called as dystrophin. And this is the germline defect in this gene often causes Duchenne muscular dystrophy or Baker muscular dystrophy. So, this will ultimately lead to the progression in muscle degeneration. So, between DMD and BMD, the same gene is the causative gene for both of them. The difference is in the type of mutation, where DMD has the frameshift mutation, whereas Becker muscular dystrophy has the missense mutation, which is less severe. So patients see the effects at the later years of their life, around 10 to 20 years, whereas DMD is seen at the early age, around 4 to 6 years of age. They can see the symptoms for the disease. So now this disease is incidence is for this particular disorder is seen 1 in 3,000 to around 5,000 males birth. Uh, Talking about the DMD and BMD, they are X-linked recessive disorders, and they cause the exonic deletion. Uh, Approximately 60 to 70% deletion, exonic deletion causes DMD or BMD, and also duplications, approximately 5 to 10%. They cause the CNVs. So, and also 20% part is for the point mutations here. So as you can see here on the right side, we have solutions that, as again I discussed this earlier, P034 and 35. They together contain around, not around, they contain all 79 exons of DMD. With this, we can ensure that the rarest deletions are also caught. And here you can see P035 has the added value because it checks for the alternative promoter in exon 1, which is DP427C. So if we have to think about the diagnostic algorithm of this, then it starts with a clinical suspicion of this particular disorder, and where the creatine kinase levels are checked, which are elevated in DMD more, lesser in BMD, Becker muscular dystrophy. Second step is always MLPA, for obvious reasons, because 80% of the cases, or 70 to 80% of the cases, they are because of the deletions and duplications. Now, although CNV stands as a major point, copy number variation for DMD disorder, there are some point mutations, as we said, around 20%, which cannot be detected by MLPA, and hence it is recommended to use this assay in combination with sequence analysis. Okay, now we'll move to the next part of the presentation, which is the spinal muscular atrophy and its global awareness. So spinal muscular atrophy is the group of autosomal recessive neuromuscular disorder which are characterized by the degeneration of the anterior horn cells of the spinal cord. Now this leads to the symmetrical muscle weakness and the atrophy, as you can see here. SMA Awareness Month is August, where the global initiative is to raise the awareness for SMA, and it's a leading genetic cause for the infant mortality. So looking at the diagnostic challenge, 95% of the SMA cases are caused by the homozygous deletion of exon 7 in the SMN1 gene. But here, the SMN2 gene also plays an important role because it determines the severity of the SMA condition. So copy numbers of SMA2 are also important. So here, the highly similar pseudo gene, which is SMN2, it makes the standard sequencing difficult for its precise copy number quantification. So NGS may not be able to quantify exact copies. So what we provide here with MRC Holland, as you can see, P021 and 22, these both kits, they are recognized as gold standard for reliably quantifying SMN1 and SMN2 copy numbers. We also have P460 ProbeMix kit. It is for the silent carrier testing, the case where both the copies of SMN gene are present on the same chromosome. Then there is another kit which is MC002. Now this kit is based on the MELT assay principle. So it is a MELT assay kit and it is for the screening of SMN1, SMN gene for the newborns. So this is the solution for SMA from MRC Holland. It also helps in the precision for therapy because accurate SMN2 copy number counting is very critical to determine the eligibility of modern gene therapies like Zolgensma and Spinraza. Talking about again NPRD 2000 impact here, SMA is grouped in group 3 disorder in India in that policy. So a definitive molecular diagnosis via MLPA is the legal gateway to access the financial support for the treatment. We'll now talk about the Prader-Willi syndrome, which dictates the epigenetic modification. So Prader-Willi and Angelman syndrome, they are distinct neurogenetic disorders. So we have this kit, which is ME028 MSMLPA. It confirms the Prader-Willi and Angelman syndromes by simultaneously analyzing copy number and methylation for the same patient. The genes which it targets are SNRPN and MEGL2 regions. It is to detect the chromosomal deletions, also to detect the uniparental disomy where both the chromosomes are from the same parent, and also imprinting center defects. Here, as I already said, it's a dual detection, CNV plus imprinting abnormalities. Looking at the molecular basis of Prader-Willi syndrome, around 70% of this disease is paternal deletion in 15q11, q13 region. 25 to 30% is maternal uniparental disomy, and around 1 to 3% is accounts for imprinting center or epigenetic defects for Prader-Willi and Angelman syndrome. Again, this SALSA MS MLPA assay, it provides you CNV and methylation together in single reaction. Talking about the diagnostic utility, again, this is a disease-specific methylation signature which is associated with Prader-Willi with a very high molecular sensitivity, and it is suitable as primary confirmatory test for suspected Prader-Willi cases. So what are the technical advantages? Here, it utilizes the methylation sensitive restriction enzyme, which is HAL enzyme for the digestion. Now, this differentiates active and silent alleles without using bisulfite conversion. So with not using bisulfite conversion, we are simply preserving the DNA's integrity. Okay. Now, this particular product is the latest launch from MRC Holland, and this is based on the technology which is digital MLPA. So let me tell you what digital MLPA is. So this NXTIC assay works on digital MLPA, as in it combines the copy number variation confidence that MLPA brings with the next generation sequencing platform. So here you were able to identify up to 60 targets in single reaction in MLPA. Digital MLPA enables you to identify up to 1,000 targets in single reaction or more than that. So D024, this is the carrier panel. It delivers an unparalleled detection of copy number variations which are associated with 10 autosomal recessive or X-linked disorders, with an additional coverage of 30 relevant single nucleotide variants. So this, again, as I said, it brings the copy number analysis into the NGS-based workflow, even for the complex genomic regions. So how do you do the analysis? Again, just like MLPA has the automated free software, it also has the automated free software. You just need to load the files, and Cofluizer Digital MLPA will give you the QC data and the reports which are ready to analyze. This is the panel coverage mentioned on the screen, the 10 autosomal recessive disorders that I mentioned, SMA, DMD, alpha beta thal, cystic fibrosis, and so on. Talking about the rare disease diagnostic solutions and its integration with cytogenetics and molecular visualization. So now integrated disease testing portfolio. So this reflects that from DSS Imagetech. It starts with the fish probe solutions we have from Abbott molecular devices, detection of chromosomal aneuploidies, microdeletion syndromes, gene-specific abnormalities is there. Also, we have advanced cytogenetic imaging with the ASI, which is Applied Spectral Imaging Platform, and karyotype analysis. We have ADS Biotech, which provides automatic cytogenetics platform such as MetaFest spreader, G banding platforms. So where these all solutions are helpful. So they are helpful in the areas such as developmental and congenital genetic disorders, microdeletion syndromes and diagnosis, also prenatal and postnatal cytogenetic evaluation. If we talk about the strategic focus in India, empowering the rare diseases with this NPRD policy and the solutions that we just discussed. So this NPRD policy has identified around 63 rare diseases for the targeted financial support. Group 1 is one-time curative treatment, which for diseases like lysosomal storage disorders, which involves stem cell transplants. Then group 2 is long-term management by a specialized diet or low-cost therapy. And group 3 is high-cost lifelong therapies, such as for DMD, SMA, where the gene therapy is involved. So, talking about what DSS via MRC Holland can provide for all these groups and their disorders. So, group 1, which has, for example, diseases such as Gaucher, we have this product P338. For Fabry, we have P159. In group 2, we have kits such as P55 for PKU. For galactosemia, we have P156 and so on. And in group 3, there is DMD and SMA, which I already spoke about. So there is also strategic coverage. There are additional solutions such as HUS and cystinosis. So we have kits for that also. So why we are discussing this? Because our goal is to move from diagnosis to excellence in the treatment. So this is the flyer which we have prepared based on the rare diseases which were mentioned in National Rare Disease Policy, for which the treatments have been allotted. So these 63 rare diseases which were mentioned, and these are the solutions from MRC Hall. I've just prepared the table which is categorizing all three groups, not mention all the diseases here. Just for example, group 1 has all these diseases. This is the MLPA solution and its clinical significance. Likewise for group 2 and group 3. You can see in group 1 there's Gaucher disease. Group 2 has Wilson disease. Group 3 has SMA, cystinosis. So we'll talk about why the tiered testing workflow works in India. With the tiered testing, we can really improve the cost effectiveness here because we have very cost sensitive healthcare environment. Also in Indian population, like a very high population, there is high pediatric burden also. And generally, copy number variation heavy disorders are very common here in India. Also, the infrastructure which is required for MLPA, which is just the thermocycler and a capillary electrophoresis system, it is mostly available in different labs across India. What clinical and research impact it makes? So this definitely reduces the diagnostic delay, which is generally seen. Then it improves the family planning. It also supports the national policy for the rare diseases, and it also enables the gene discovery, via collaborative networks. Okay. Now, rare disease is entering into curative era because here we can see from diagnosis to disease modification. Gene therapy is moving the rare diseases from supportive care towards a very potentially transformative one-time treatment. And there are approved therapies for SMA and also inherited retinal disorders. It proves the precision medicine is now actually a clinical reality. Talking about the molecular testing, which are the real gateways to the therapy, as I already said, genetic confirmation is mandatory from the from the center of excellence for one to get the funds for the treatment. So here, NGS detects SNVs. We have exon level deletions also, and many other variants which are detected. So here, precision diagnostics is ensured. How? Because once we find out the right variant, right patient is identified with the right variant, and which will lead to the right therapy at as early as possible speed. So the reality check, regulatory momentum currently is very strong. US FDA has is advancing rare gene therapy approvals recently, and the next challenge will remain translating these innovations which are happening into real access, managing or balancing the cost, making availability of infrastructure, and also the long-term durability. So these are certain news which were published. So first news is about the success story, which is the gene therapy for Hunter syndrome. So there is this kid Oliver Chu. He has made the medical history as the first person to receive the pioneering gene therapy for Hunter syndrome. It's a rare condition. Often it is called as childhood dementia. And after receiving the groundbreaking treatment in Manchester, Oliver is now thriving and meeting developmental milestones that were previously thought impossible. So another success story is, or rather, breaking news is about Japan. Japan has debuted as the first gene therapy for Duchenne muscular dystrophy. So they have officially launched this Elevidys, the first regenerative gene therapy for DMD. And now this therapy is covered by the National Health Insurance. This one-time infusion targets the disease root cause by restoring the dystrophin function before the muscle loss becomes irreversible. Now, this is one of the story about resilient spirit, which goes beyond the diagnosis. Actually, this student Vihan Gupta, he scored 93.8% in CBSE exam. He proved that while rare diseases like DMD, they may limit the body, but they cannot be bound. They cannot bind or they cannot be bound to a determined mind. So again, this illustrates us the current reality of living with the rare disease such as DMD and the importance of social and educational support. Another clinical success, which is again not so old, it's a recent one only, for the severe CPS1 deficiency. In the medical first, an infant with the CPS1 deficiency was successfully treated with the personalized CRISPR therapy designed to repair his specific CPS1 gene mutation. This news illustrates us the future potential of medical intervention and bespoke medicines, the personalized ones. So now these are the key take-home messages from this entire presentation. First is global and national impact. Rare diseases collectively affect around 300 million people globally and 70 to 96 million people in India. The main aim is to end the odyssey, the diagnostic odyssey. Definitive molecular diagnosis is the only way to end this 5 to 7 years of diagnostic odyssey. About the legal gateway, under India's NPRD 2021, a confirmed genetic report from designated center of excellence is a mandatory legal requirement to access up to 50 lakhs in financial treatment support. About tier testing, no single platform is suitable to detect all variants. Integrating technologies such as MLPA, which is gold standard for CNV, is critical for accurate results. About curative future, we are moving from supportive care to a curative era of gene therapies and personalized CRISPR medicines. Talking about beyond the medicine part, social and educational support is very important in this field because a diagnosis does not define the patient's actual potential. So social and educational awareness in rare diseases become very important. So I'm done with my presentation. Thank you, everyone. If anyone has any question, please let me know.